Imagine an animal whose DNA is so unlike anything else on Earth that it challenges everything scientists thought they knew about genetics and evolution. Octopuses, known for their intelligence and camouflage, possess a genetic code far more complex and mysterious than most species, with over 33,000 protein-coding genes, more than humans. This remarkable genetic uniqueness opens up new frontiers in biology and medicine, raising questions about how life can diversify in unexpected ways. In this video, you will discover the extraordinary features of octopus DNA, how it compares to other species, and what it means for the future of scientific research. Prepare to explore a story that rewrites the rules of life's blueprint and reveals secrets hidden beneath the waves. Subscribe and watch until the end. This journey into the unknown will leave you amazed. Octopuses are among the most fascinating creatures in the ocean, known for their intelligence, camouflage abilities, and unique anatomy. But what truly sets them apart begins at the most fundamental level, their DNA. Unlike many other species on our planet, octopus DNA shows characteristics that challenge our usual understanding of genetics and evolution. To appreciate why octopus DNA is so unique, it's important to first understand what DNA typically looks like in most animals and then see how octopuses differ. DNA, or deoxyribonucleic acid, is the biological blueprint that dictates how living organisms grow, develop, and function. It is composed of sequences of 4 chemical bases, adenine, thymine, cytosine, and guanine, that pair up to form the famous double helix structure. These sequences encode the instructions for building proteins, which perform most of the work in cells. In most animals, including humans, DNA is organized into chromosomes inside the cell nucleus, and each species has a specific number and arrangement of these chromosomes. For example, humans have 46 chromosomes, while fruit flies have 8. Octopuses, however, break many of these norms. One of the most notable differences is the sheer size and complexity of their genome. The genome is the complete set of genetic material in an organism. The common octopus, Octopus vulgaris, has an estimated genome size of about 2.7 billion base pairs. This number alone might seem ordinary compared to humans, who have roughly 3 billion base pairs. But what makes octopus DNA remarkable is not just its size, but its composition and organization. The octopus genome contains a large number of genes that are either unique or have been extensively modified compared to other animals. Researchers have discovered that octopuses possess an unusually high number of genes related to neural development, which may explain their advanced nervous system and problem-solving abilities. For instance, genes involved in synapse formation and neurotransmitter regulation are particularly abundant. This genetic richness supports the octopus's capability for learning, memory, and even tool use, traits that are rare among invertebrates. Another striking feature of octopus DNA is the presence of what scientists call gene family expansions. This means certain groups of genes have multiplied extensively, creating many copies with slight variations. One example is the protocadherin gene family, which is involved in building the complex wiring of the nervous system. Octopuses have more protocadherin genes than many vertebrates, including humans. This expansion likely contributes to the sophisticated brain structure of the octopus, which is divided into several lobes with specialized functions. Beyond gene content, octopus DNA also displays unusual patterns of RNA editing. RNA is the intermediate molecule that carries genetic information from DNA to the protein-making machinery in cells. In most animals, the RNA sequence closely matches the DNA template. Octopuses, however, extensively edit their RNA, altering many nucleotide bases after transcription. This process allows them to diversify their proteins without changing their underlying DNA sequence, providing a flexible way to adapt to environmental changes rapidly. For example, RNA editing can modify proteins involved in neural signaling, potentially fine-tuning brain function in response to different conditions. Octopuses also exhibit a unique arrangement of their DNA in their chromosomes. While most animals have relatively stable chromosome structures, octopus chromosomes contain large sections of repetitive DNA and transposable elements, sometimes called jumping genes. These elements can move around within the genome, causing mutations and genomic rearrangements. Such activity might contribute to the genetic diversity and adaptability seen in octopus populations. One more point worth noting is that octopus DNA challenges the traditional boundaries between vertebrates and invertebrates in terms of complexity. Despite being invertebrates without a backbone, octopuses have evolved genetic features that rival or even exceed those found in many vertebrates. Their genome is a testament to the diverse paths evolution can take, especially in marine environments that impose unique selective pressures. In summary, octopus DNA is unlike that of any other species because it combines a large and complex genome with unique gene expansions, extensive RNA editing, and dynamic chromosome structures. These genetic traits underpin the remarkable capabilities of octopuses, from their intelligence to their adaptability. Understanding octopus DNA not only sheds light on their biology, but also broadens our perspective on how genetic information can be organized and utilized in living organisms. As research continues, we may discover even more surprising features of octopus genetics, further highlighting the intricate tapestry of life on our planet. When we think about DNA, we often picture a long, twisted ladder made of 4 basic building blocks: adenine, thymine, cytosine, and guanine, arranged in a double helix. This structure carries the genetic instructions for almost all living organisms on Earth. However, not all DNA is created equal, and the octopus is a remarkable example of this. Its genetic makeup reveals some fascinating differences that set it apart from virtually every other species we know. Octopuses belong to a class of mollusks called cephalopods, which also include squids and cuttlefish. Despite being soft-bodied creatures without bones, they exhibit astonishing intelligence and adaptability. Much of this can be traced back to their unique genetic features. One of the most striking aspects of octopus DNA is the sheer size and complexity of their genome. For example, the common octopus has around 2.7 billion base pairs, comparable to humans in size, but with a different organization and content. Unlike many animals, octopus genomes contain an unusually high number of genes related to neural development and signaling. This abundance helps explain their sophisticated nervous systems and problem-solving abilities. To put this into perspective, octopuses have roughly 33,000 protein-coding genes, which is far more than humans, who have about 20,000 to 25,000. This expanded gene repertoire likely contributes to their complex behaviors, such as using tools, navigating mazes, and exhibiting individual personalities. Another distinctive feature lies in how octopuses edit their RNA, the molecular messenger that carries instructions from DNA to the cell's protein-making machinery. While RNA editing occurs in many organisms, octopuses do it at an exceptionally high level, especially in their nervous tissues. This means they can effectively alter their genetic instructions after transcription, creating protein variants without changing the underlying DNA sequence. It's akin to having a versatile toolkit that can be customized on the fly, to meet different environmental challenges or developmental needs. Moreover, octopuses display an intriguing pattern of gene rearrangement. Some of their genes responsible for brain function have undergone duplications and rearrangements not commonly seen in other animals. This genetic shuffling may provide the raw material for the evolution of novel traits that underpin their intelligence and adaptability. It's a bit like remixing a song to create an entirely new version with a different sound, rather than just repeating the original track. One might wonder how octopus DNA compares to more familiar animals like vertebrates. Despite having a complex nervous system and advanced behaviors, octopuses evolved along a very different path. Their lineage diverged from ours about 600 million years ago, leading to a genome that reflects unique evolutionary pressures. For example, octopus genes involved in the development of their arms and suckers show expansions and modifications that enable intricate motor control and sensory perception, features critical for their survival in diverse marine environments. Interestingly, octopus DNA lacks certain genes found in vertebrates, such as those responsible for some aspects of the immune system, but compensates through alternative mechanisms. This highlights how evolution doesn't always follow a single blueprint. Instead, it can arrive at similar outcomes through different genetic routes. The octopus genome demonstrates how life can innovate in unexpected ways, creating complexity without following the familiar genetic patterns observed in mammals, birds, or fish. In practical terms, these unique genetic features have made octopuses a subject of intense scientific interest. Researchers study their genomes to better understand the molecular basis of intelligence, neural plasticity, and even regenerative abilities. They can regrow lost arms thanks to specialized genetic programs. Insights gleaned from octopus DNA may one day influence fields ranging from neuroscience to biotechnology, offering clues about how to design adaptable systems or develop new therapeutic strategies. In summary, the octopus genome is a powerful example of nature's creativity. Its large and complex genetic makeup, extensive RNA editing, gene rearrangements, and unique evolutionary path combine to produce an animal that challenges our assumptions about intelligence and biological design. For anyone curious about the diversity of life on Earth, the octopus serves as a reminder that behind every creature lies a genetic story, full of surprises and lessons waiting to be discovered. Understanding these unique features not only deepens our appreciation of octopuses, but also broadens our perspective on the possibilities encoded within DNA itself. When we look at the DNA of an octopus and compare it to the DNA of other species, we uncover some fascinating differences that highlight just how unique these creatures are. DNA, as you might know, is the blueprint for all living organisms, containing instructions that tell cells how to function and grow. While all life on Earth shares the basic chemical structure of DNA, the specific sequences and the way that DNA is organized can vary widely among species. And this variation is where the octopus stands apart. One of the most striking things about octopus DNA is its sheer complexity. Octopuses belong to a group of animals called cephalopods, which also includes squid and cuttlefish. Unlike many other animals, the octopus genome is relatively large. In fact, the genome of the common octopus contains about 2.7 billion base pairs, which is roughly as much DNA as some mammals, like humans. However, it's not just the size that is interesting, but the content and organization of that DNA. When scientists first sequenced the octopus genome, they found that a large portion of their DNA consists of what are called transposable elements, sometimes referred to as jumping genes. These sequences can move around within the genome, and this mobility is believed to contribute to genetic diversity and adaptability. While transposable elements are found in many organisms, the octopus genome has an unusually high number of them, suggesting that this feature may play a key role in the octopus's ability to learn, adapt, and exhibit complex behaviors. Another notable difference is the number and variety of genes related to neural development and function. Octopuses are known for their advanced nervous systems, which allow them to solve puzzles, navigate mazes, and even use tools. This is unusual for invertebrates, which typically have much simpler nervous systems. In the octopus genome, researchers have identified a large expansion of certain gene families associated with synaptic function, which are essential for communication between nerve cells. This genetic expansion likely supports the octopus's remarkable intelligence and sensory abilities. Interestingly, octopus DNA also shows unique patterns when it comes to RNA editing. RNA is a molecule that helps translate DNA instructions into proteins, and in most animals, RNA sequences closely match the DNA template. However, in octopuses, there is an extraordinary level of RNA editing, particularly in genes involved in neural function. This means that even after the DNA blueprint is set, octopuses can tweak their RNA molecules, effectively fine-tuning protein function on the fly. This capacity adds a layer of flexibility to their biology that is rare or absent in most other animals. Comparing this to other species helps put it into perspective. For instance, mammals like humans have large and complex genomes too, but they do not exhibit the same level of RNA editing seen in octopuses. On the other hand, simpler animals such as fruit flies or nematodes have much smaller genomes and less genetic complexity, which corresponds to their simpler nervous systems and behaviors. The octopus sits somewhere in between, but boasts unique features that make it stand out. It's also worth mentioning how octopus DNA differs structurally. Unlike vertebrates, where genes are often tightly packed, octopus genes are more spread out and interspersed with non-coding regions. These non-coding sequences were once thought to be junk DNA, but now scientists understand they can regulate gene expression and contribute to genetic innovation. The octopus's genome organization might therefore provide more opportunities for gene regulation and innovation, supporting their adaptability and complex traits. Overall, the comparison between octopus DNA and that of other species highlights how diverse life can be at a molecular level. The octopus genome challenges the traditional view that complexity of behavior correlates directly with genome size or gene count. Instead, it shows that genetic mechanisms like transposable elements, RNA editing, and gene family expansions can create complexity in new and unexpected ways. In practical terms, understanding these differences is important not only for biology and evolution studies, but also for biotechnology and medicine. Studying how octopuses edit their RNA or regulate their genes could inspire new approaches to treating neurological diseases or developing adaptive technologies. In summary, while all species share the fundamental blueprint of DNA, the octopus uses this blueprint in ways that are quite different from most animals. Its genome size, content, structure, and the unusual genetic mechanisms it employs all contribute to the remarkable adaptability and intelligence we observe in these fascinating creatures. By studying these differences, we gain insight not only into the octopus itself, but also into the broader possibilities of life's genetic diversity. When we dive into the world of octopus genetics, what stands out most is how uniquely complex their DNA is compared to other species on Earth. This uniqueness isn't just a curious quirk, it has significant implications for science, medicine, and even our understanding of evolution. To appreciate these implications, it's important to explore what makes their genetic makeup so distinct and why that matters beyond just being an interesting fact. Octopuses have a remarkably large genome with around 33,000 protein-coding genes, far more than humans, who have about 20,000 to 25,000. This abundance of genes offers the octopus an incredible range of potential biological functions. One of the most notable features of their DNA is the presence of extensive RNA editing. Unlike most animals, where DNA essentially dictates the blueprint, octopuses can alter their RNA messages significantly after transcription, effectively changing the proteins they produce without altering the underlying DNA sequence. This allows them to adapt more rapidly to changing environments by fine-tuning their physiology in real time. For example, they can adjust proteins in their nervous system to cope with different temperatures or other environmental stresses. This ability is somewhat analogous to having a flexible instruction manual that you can revise on the fly, rather than being stuck with a fixed set of directions. This genetic flexibility has important implications for neuroscience and biomedical research. Octopuses are known for their advanced cognitive abilities, such as problem-solving and learning, despite being invertebrates. Understanding how their unique RNA editing contributes to neural complexity could provide insights into brain function and neuroplasticity in general. It opens up avenues for studying how gene expression can be regulated beyond the static DNA code, potentially inspiring new treatments for neurological diseases where gene expression is disrupted. Furthermore, the octopus genome contains many genes that are similar to those found in vertebrates, despite the two groups being evolutionarily distant. This phenomenon, called convergent evolution, suggests that similar genetic solutions can arise independently in unrelated species to solve comparable biological challenges. It challenges the straight-line perspective of evolution and highlights the versatility and inventiveness of life's genetic toolkit. From a practical standpoint, these parallels make octopuses a valuable comparative model for studying gene function and evolution, shedding light on fundamental biological processes that are shared across diverse life forms. Another critical implication of their genetic uniqueness relates to biodiversity and conservation. As we learn more about the genetic makeup of octopuses, it becomes clear that their populations are more genetically diverse than previously thought. This diversity is crucial for their ability to adapt to rapidly changing ocean conditions, such as warming waters and pollution. Protecting the habitats and genetic diversity of octopuses isn't just about preserving a single species. It's about maintaining the resilience of marine ecosystems that rely on their ecological roles, from controlling prey populations to recycling nutrients. In addition, the octopus genome includes genes that produce novel proteins not found in other animals. These unique proteins could have applications in biotechnology and medicine. For instance, they might inspire new biomaterials or pharmaceuticals with properties adapted to marine environments, such as resistance to extreme pressure or salinity. Studying these proteins can broaden our understanding of protein structure and function, potentially leading to innovative technological advances. To sum up, the genetic uniqueness of octopuses isn't merely an interesting scientific fact. It has profound implications that extend into multiple fields. Their flexible RNA editing challenges our conventional understanding of genetics, offering new perspectives on adaptability and gene regulation. Their evolutionary parallels with vertebrates provide valuable insights into biology and evolution. Their genetic diversity underlines the importance of conservation efforts, while their novel genes offer promising avenues for future biotechnological innovations. Understanding octopus DNA enriches not only our knowledge of one of the ocean's most fascinating creatures, but also expands our horizons in science, medicine, and environmental stewardship. As we have explored in previous chapters, the octopus's DNA presents a fascinating puzzle that challenges many of our assumptions about genetics and evolution. Scientists have uncovered remarkable features in its genome, from the vast number of genes to unique arrangements that differ significantly from other animals. But what lies ahead in the study of octopus DNA? How might future research deepen our understanding of this extraordinary creature and its place in the natural world? One promising avenue for future discoveries involves the continuing refinement of genomic technologies. As sequencing methods become faster, cheaper, and more accurate, researchers will be able to delve into the octopus genome at an even finer scale. This will allow them to identify subtle variations and regulatory elements that control gene expression. Understanding these mechanisms will shed light on how the octopus develops its complex nervous system and remarkable behaviors, such as problem-solving and camouflage. Moreover, advances in comparative genomics will enable scientists to explore the DNA of other cephalopods and related species. By comparing these genomes, they can trace the evolutionary history that led to the octopus's unique genetic features. This will help clarify which genetic traits are shared among cephalopods and which are exclusive to octopuses, revealing the molecular basis for their distinctive adaptations. Another exciting prospect is the application of gene editing tools like CRISPR to octopus DNA. Although still in early stages, such techniques could allow researchers to experimentally test the function of specific genes. For example, by selectively activating or deactivating certain genes, scientists might observe how these changes affect the octopus's nervous system development or behavior. This experimental approach has the potential to unlock many secrets about the relationship between genes and complex traits in cephalopods. In addition to laboratory studies, field research will continue to play a critical role. Octopuses live in diverse marine environments, and their genetic responses to different habitats and ecological pressures remain largely unexplored. Future studies may investigate how environmental factors influence gene expression and adaptation, providing valuable insights into how these animals cope with changing ocean conditions. Such research is particularly important in the context of climate change and human impacts on marine ecosystems. Furthermore, interdisciplinary collaboration between geneticists, neuroscientists, marine biologists, and computer scientists will enhance our ability to interpret octopus DNA data. For instance, computational modeling and artificial intelligence can help analyze large genomic datasets to identify patterns that might be missed by traditional methods. Integrating diverse expertise will accelerate discoveries and generate more comprehensive models of how octopus biology operates at the molecular level. Looking beyond scientific knowledge, these insights have practical implications as well. Understanding octopus genetics could inspire innovations in biotechnology, such as developing new materials based on the octopus's skin properties or novel neural networks inspired by its brain structure. Additionally, conserving octopus species and their habitats depends on detailed genetic information to assess population health and resilience. In summary, the future of octopus DNA research is full of potential and challenges. With advancing technologies and collaborative efforts, we can expect to uncover deeper layers of genetic complexity and evolutionary history. These discoveries will not only enrich our understanding of one of the most intriguing creatures on Earth, but also contribute to broader scientific and environmental goals. As we continue to explore the genetic code of the octopus, we are reminded of the vast diversity of life and the many mysteries still waiting to be solved beneath the waves.